going to start off with a talk by Adam Frankish with some time for questions. Then we're going to do a hands-on first demo with Sarah Donaldson. Then I'm going to give a talk about some of our latest projects, and then we have a final demo from Toby. The slides are all available on the Ensemble training website, and I will put that up into the Slack channel as well. So do you want to get started, please, Adam?
Absolutely, thanks, Jane. My name is Adam Frankish. I lead the Ensemble HAVANA gene annotation team at EBI, and we play a role in the annotation of genes for the Gencode project. So the overall goal of Gencode is to annotate all evidence-based gene features. So this means all features in protein coding genes, long non-coding RNAs, small RNAs, and pseudogenes in the human and mouse genomes with high accuracy, and then to release these annotations so people can use them in their own analysis and research. And there are two things that we know about the gene set that we annotate. One is that it's incomplete. So this sets our goal of annotating all gene features as quite a challenging one. We know that from large RNA-seq and long transcriptome data studies that we don't have all genes or all transcripts annotated yet, but we're working towards that. And we also know that every error in gene annotation can potentially affect any downstream analysis done with that gene annotation, including potentially in clinical diagnosis. And so this is why we're very, very rigorous in trying to maintain very, very high accuracy, high specificity, essentially, in the annotation that we produce. So the Gencode consortium consists of 7 groups, and I've organised them in this way as a sort of hub and spoke sort of arrangement. Sitting at the centre are the gene annotation team at EBI. We produce the gene annotation that goes into the Gencode gene set, but we're supported through bioinformatic analysis, pipelines and tools, and by primary data generation by the other 6 teams within the consortium. So, for example, Manolis Kellis's group at MIT provides us support with comparative genomic analysis, including the PhyloCSF tool that they created and maintain. Benedict Payton's group at UCSC again provides us with a lot of bioinformatics support and analysis, and again work with comparative genomics, including whole genome alignments using the Cactus tool. The Michael Tress's group at CNIO provide us with protein bioinformatics support, including producing a tool called APRIS, which tries to identify the most likely principal coding isoform at every locus that we annotate in human and mouse, and his work has been extended to some other species where there is data available. Mark Gerstein's group at Yale provide us with support for support and analysis in annotating pseudogenes. And this not only they've not only developed pipelines to help us with discovery of pseudogenes in human and mouse genomes, but also in characterization of pseudogenes, specifically with looking at their expression and other features that could suggest functionality. And then the 2 groups at the base here are Jyoti Chowdhury's group at the Institute of Cancer Research in London and Rodrigo's group at CRG in Barcelona. And they provide us with primary experimental data. ICR give us proteomics data, and CRG provide transcriptomic data. But in addition to that, they both provide a large amount of analysis tools and that is both generalized and specific to helping us with gene annotation. And as I say, the reason why I put us in the center of the wheel here is that all these groups and all the tools and analysis and data that these groups produce supports the annotation team in producing better quality gene annotation. And in addition to that, the groups also have quite a lot of contact with each other directly within Gencode. So, for example, the group at UCSC are producing better alignments, which feeds into the PhyloCSF tool, which uses whole genome alignment data to identify protein coding regions. And then that's passed on to the annotation teams, and as well as the sort of the direct contacts as part of the Gencode project, they also have contacts and collaborations outside Gencode. Sort of the relationship I've highlighted at the bottom between Yale, ICR, and CRG all these teams are working together on the NTEX project, which is a sort of ENCODE GTEX collaboration. The Gencode is the reference gene annotation you will see if you, for human and mouse, if you go to the Ensembl or UCSC genome browser. So obviously in both you're able to load other annotation sets such as RefSeq, but the one that you will see as a default is the Gencode annotation. And Gencode is used as a reference by many large genomic studies, both those studying genome biology and clinical consortia that use our annotation as the basis for downstream analysis. And obviously ENCODE is one of the notable ones in there. So coming back to what we do in our annotation group, essentially annotators are doing two things. We provide structural annotation, or what we call structural annotation, which is determining the correct gene structure of the features that we annotate. So primarily, this is generally looking at aligned transcriptomic data, but can use other data types as well to identify the start of a transcript, the splice junctions at the end of a transcript. And as I said, historically, when we started doing this work, we were using cDNAs and ESTs. Obviously, people no longer produce those anymore. So we've moved on now to using RNA-seq data and also long transcriptomic data, such as that produced by PacBio and Oxford Nanopore. And the second thing that we do in annotation is what we call functional annotation. So we take the transcript models that we've correctly annotated, and then we try and figure out what they do. Are we annotating a protein coding locus or a long non-coding RNA or another non-coding RNA gene, or are we annotating a pseudogene of a protein coding gene? And the way that we can do this in many different ways. We can look at e.g. aligned protein data from UniProt. We can look at conservation, cross-species conservation data. We can look at many different kinds of comparative genomic data. We can look at primary experimental data from mass spec experiments or ribosome profiling experiments, and where there is literature on the gene that we're investigating, we can go and read papers and try and figure out how to best represent what they've identified in the annotation we produce. So as I say, this is just sort of recapitulating what I've just said, is that we sort of essentially start off with aligned transcriptomic data. All our annotation is aligned to the reference human genome at the moment, and the reference mouse genome, and then we layer on pretty much as many other types of data that we need to be able to correctly represent the transcript structures and their functional potential. So in addition to identifying the functions of a gene at a sort of a higher level, within the gene annotation, we identify, we try and give some sort of functional characterization of the transcripts within that gene. So for example, this protein coding gene, we annotate protein coding transcripts that we think at least have the potential to encode functional proteins. And these are represented here in the CDS is represented as the open green boxes, and the 5' and 3' UTRs as the filled red boxes. We also identify transcript models that contain a premature stop codon, which we think is likely to lead them to be targeted by the nonsense-mediated decay, or NMD pathway. Here the CDS is represented as an open purple box, and again the UTRs are in red. We annotate CDSs in these genes because we know that they undergo at least one round of translation, and so you know, in proteomics and other experiments, it's been demonstrated that they can produce protein. And we also annotate transcripts that show intron retention. Again, I take you back to my first slide. Our intention is to represent all sort of genic features, and so we are at the moment capturing retained introns, but making sure that we label them as correctly as we possibly can, so they don't get drawn into the potential pool of protein coding transcripts. For our long non-coding RNAs, historically we've annotated long non-coding RNAs with sort of relationship to their geography or geographical position to a protein coding gene. So for example, intergenic, genes that long non-coding RNAs that lie between protein coding genes fall into our long intergenic non-coding RNA class. genes that overlap protein coding genes on the opposite strand fall into our antisense class. Genes that lie within introns of protein coding genes are called sense intronic, and those that for which protein coding genes lie in the intron of the long non-coding RNA are called sense overlapping. Now we will maintain these biotypes in the files that we release, so people can still filter by them if they wish to do so. But we're moving towards a more I guess sort of biologically focused or sort of data-driven labeling system, where we hope to be able to tag transcripts to give more information about the regulatory elements that are driving their transcription. So we'll be able to say whether they are whether their transcription is driven from a promoter, is that promoter shared on the opposite strand with the protein coding gene, for example, or does their transcription derive from enhancer activity. Another class of transcripts that we annotate are pseudogene loci. Obviously, what we've tried to capture with pseudogenes is how they were made. So, for example, pseudogenes that were derived via retrotransposition events and reinsertion into the genome are tagged as process pseudogenes. Pseudogenes that have arisen via genome duplication events are called duplicated pseudogenes or unprocessed pseudogenes. We also annotate unitary pseudogenes, which are ancestral protein coding genes that have picked up a fixed disabling mutation in the human lineage or earlier, but they do have clear evidence of ancestral coding potential. And we have what we call polymorphic pseudogenes currently, which aren't really pseudogenes. They are protein coding genes in other individuals, but they have an allele that contains a disabling mutation in the reference genome. So this is very much sort of derives from the reference centric nature of the annotation as it has been to date. This is something that we may be looking to change in future, but we'll make it clear where the polymorphic where transcripts or genes formerly tagged as polymorphic pseudogenes are if they're reassigned to different functional classes later on. So we release our annotation regularly, approximately 4 times a year. It coincides with ensemble releases. Although the Gencode annotation is available from the URL that you can see below, gencodegenes.org. You can download the files from there too. Just to note a couple of things. This is Gencode 35, which is our current release and corresponds to Ensemble Release 101. We have about almost 20,000 protein coding genes in human. Although one thing to flag here is that this includes loci that read through between 2 independent protein coding loci. We're not certain about their functional significance, but we do record them and report them, and we make sure that we tag them up with remarks so that anybody using the gene set can identify where these genes come from and what their relationship to other genes is. Similarly, in mouse, we make a mouse release on a very, very similar schedule, almost identical schedule to human generally, and will do going forward. Our latest release of mouse is Gencode M25. And we have more protein coding genes in mouse than human. In fact, almost 2,000 more protein coding genes in human. Again, this number is slightly inflated by these read-through transcripts, and also maybe slightly higher because we haven't had a chance to do the same intensity of QC on the mouse protein coding gene set as we have on human, but it looks like mouse will end up with substantially more protein coding genes than human, even once this is complete. An important thing to note here is that we have moved our annotation to the new mouse reference assembly GRCM39. This annotation will be available in the spring of 2021, and all annotation that we do in mouse from now on will be on this new reference genome and not on GRCM38 anymore. Just going back a bit into the history of Gencode, in fact, pre-Gencode history. So back in 2003, 2004, the Gencode annotation, or the annotation that we were doing as part of the Human Genome Project, wasn't stored in the same database or even a compatible database to the gene annotation produced by the Ensemble group at the time. So in 2003, 2004, at the start of the millennium, only protein coding genes and pseudogenes were represented in the gene set. and these were all produced by automated annotation pipelines. In 2005, we produced we moved into an ensemble schema database, and so we were able to compare the annotation between the two. We can see that it's in these stages. It's sort of during this period that you can see the first long non-coding RNA genes appear in the manually annotated set, which is labeled V for the Vega database that they used to be stored in. But also the ensemble automated annotation pipelines had started annotating small RNAs, and this pattern continued over the next few years, where you can see, if you look at the columns labeled V, the sort of an increase in the gene count as we extended our coverage with the manual annotation, and also an increase in sort of some fluctuations in the gene count for the automated annotation as well. Starting in 2009, we produced our first merge set of automated and manual annotation. And I think from that point on, I think the pattern you can see is that the number of protein coding genes after an initial drop appears to have hit and the initial drop was due to essentially manual QC of previously automated gene models that shouldn't have been labeled as protein coding genes, but had been historically, to a relatively flat number of protein coding genes. The number of pseudogenes in red grew steadily over the years and has achieved some sort of stability now. The pale blue small RNAs essentially has remained relatively static. There's a noticeable decrease between 2015 and 2016 when there was an improvement to the detection algorithm that reduced the false positive rate in the small RNA predictions, and that this remains the only fully automated part of the process, although that will change going forward when we will do much more manual annotation of small RNAs. But the class that continues to grow and has been growing since 2009 and continues to grow are the long non-coding RNAs. We are still adding long non-coding RNA genes at a fairly high rate, and I think it's fair to say that this is the class of gene where we know that we have the most missing annotation in terms of the genes. As I said before, the headline rate of protein coding genes has remained relatively static, but that does hide some churn. So in almost every release, and certainly in these sort of 3 recent releases that I show here, we add some genes. In here, it's sort of, roughly 10 genes per release are new protein coding genes are added, and probably slightly more than that on average are removed, although this does fluctuate. We remove protein coding genes because we are constantly doing QC on the protein coding genes that we have annotated and assessing them to see, with the help of our Gencode partners, to see whether they have or whether they lack characteristics that we would expect in a protein coding gene. So, for example, can we find any evidence of transcription, enough significant evidence of transcription? Can we find any evidence of translation at all in if, for example, proteomic or ribosomal profiling data? Do we see any evidence of conservation that suggests protein coding potential? And so we're constantly reviewing genes to that end. We're also interested in finding new protein coding genes that aren't annotated, and one of the main ways that we've done that is using the PhyloCSF tool produced by our Gencode partners at MIT. And what PhyloCSF does is look for signatures of protein coding potential in whole genome alignments, for example, across mammals or vertebrates. And if you look at this example here in the highlighted black box, we have a protein coding exon and a protein coding gene. We can see from essentially a whole genome calculation in all 6 frames of PhyloCSF coding potential. We can see a signal, a clear peak in the PhyloCSF signal under the protein coding exon, and we process that signal to identify regions of the genome that we think have protein coding potential. And of course, this is a positive control example I'm showing you here with a known protein, an exon in a known protein coding gene. But what we then do is we then go and look at unannotated regions of the genome and look at these PhyloCSF region predictions here. And in this region of the genome, you can see that there are many potential putative protein coding regions that have been identified. These may represent separate genes. They may represent exons within another gene. They may be independent genes, or they may be unannotated features that we can connect to other known protein coding genes with transcriptomic data now that we have it. And so this gives us a set of things that we can investigate. And we've got a fairly long-running process now where we take these phylo-CSF regions, run on the whole genome for human and mouse, and we identify in both genomes, we identify roughly just over 70,000 potential protein coding regions in human and mouse. And we have gone through multiple rounds of assessing the top scoring ones, recalculating the way that the scoring is done to identify, continuously improve our ability to take the most likely protein coding regions. And this has allowed us to find new protein coding genes fairly well. We found several hundred new protein coding genes using this method. So here's an example of a gene which is now called SMIM31 in human. We didn't have anything annotated here previously, and we were led to this region by this phylo-CSF peak in the second exon of the gene. If we looked in mouse, obviously we're annotating human and mouse at the same time. We could look in mouse and we could find we actually found a long non-coding RNA that had been annotated in mouse. But when we looked at the potential CDSs that we could annotate in both these transcripts, we found that a very, very well conserved CDS was annotatable in both. And so this led us to update our annotation in both human and mouse to produce a protein coding two protein coding orthologs. And what's interesting about this, one of the reasons why we may not have detected this in the first pass of our annotation is that the RNA-seq data suggests a very, very restricted expression profile in gut. And it may just have been that at the time that we did the original annotation, we just didn't have this data available. So we also annotate alternative splicing, as I mentioned earlier, predominantly of protein coding genes and long non-coding RNAs. We annotate all varieties of alternative splicing that you can imagine, and I'll just sort of prelude what will be talked about later by saying, as I mentioned before, we annotate different classes of protein coding gene, but I think one of the protein coding or different classes of transcript within protein coding genes. But one of the important things I want to mention here is that in addition to having full-length transcripts, you can see those with 5' and 3' UTRs in red. We also annotate partial transcripts. If the transcriptomic data that we're using to support the model that we're annotating doesn't extend to the full length of the transcript, to the full length of the locus, we don't try and connect things together to make something that's full length when there's no support for it. Because what we're trying to do here is be very careful and specific and not introduce transcripts to the gene set that might not exist in biology. So we try as far as we can not to be predictive about this, but only extend our models as far as the data permits. And because we're making so many transcripts these days, we for a while have had this classification of transcripts as into a basic set and a comprehensive set. The basic set are essentially, for protein coding genes in this illustration, are full-length transcripts with a CDS start and a CDS end annotated, whereas the comprehensive set is essentially all the other transcripts. So transcripts that are given the NMD biotype, which do have CDS start and CDS end, will not be part of the basic set. They'll be part of the comprehensive set. And similarly, all those truncated partial transcript models that I've just mentioned are part of the comprehensive set. So if you want to see a particular splice form, you may be able to find it in the comprehensive set, but it won't be in the more restricted basic set. Sorry, I'm losing my animation. And again, just for a bit of historical perspective on this, since the sort of the start of the annotation, we saw for the first few years we saw essentially a slow and relatively steady increase in the number of transcripts that we've annotated in all gene biotypes. Through the ENCODE scale up to the whole human genome, we saw a relatively rapid increase. As you can see, the two classes of gene or two biotype of gene where we see an increase in transcript note count is in the long non-coding RNAs, and especially in the protein coding genes. We've had a period of sort of relative maintenance where we've actually been targeting genes or targeting our annotation in a more specific way. So broadly, we haven't added lots and lots of transcripts, although we hope that the transcripts we've added are important. But in 2019, you can see a kick up here, and this essentially reflects our the beginnings of our ability to integrate long transcriptomic data sets from PacBio and PacBio and Oxford Nanopore. And this graph will continue to increase at this rate, if not more steeply, as over the next few years as we integrate these sort of large numbers of high quality transcriptomic sets. And I think with that, we're going to stop for a second. Does anybody have any questions? Are there any questions that have come up that we should deal with now? I can't see the Slack chat, so sorry, the chat.
No, nothing at the moment, Adam.
Nothing at the moment.
Carry on and get. Oh, hang on, we have a question. We have a question here. So, what percentage of those pseudogenes in humans show high sequence homology between pseudogene and protein coding gene?
I can't give you a percentage, but it's a fairly large number, because pseudogenes that are obviously pseudogenes have been created essentially continuously, although there have been a couple of kind of large bursts of pseudogene activity. Processed pseudogenes often have high homology, and in fact sometimes they have very, very high homology indeed. I think we probably have some maybe not super recent analysis of exactly what the percentage homologies are like, but that has been published in the past by Mark Gerstein's group. We'll see if we can answer that in more detail in the Google Doc in the next week or so. But I don't have the figures to hand. But some pseudogenes are essentially 100% sequence identical to the parent gene. They just lack the structure. So processed pseudogenes generally have lost all their introns as they were through the splicing process before they're reinserted, or before they're copied and reinserted. And so that is what gives us the hint. Although we always have to be mindful that retrotransposition has led to the creation of some functional protein coding genes, and we have to be careful, or we try to be careful, not to automatically label them as pseudogenes where there is any evidence at all that they could be actually encoding new proteins. I don't know if that's helpful. but we'll try and put a link to references that have asked questions about similarity more generally in the Google Doc after the call's finished.
Great, thanks, Adam. Shall we move on to Sarah, who's going to do a live demo? So if you can stop sharing the screen.
Getting the deal. Okay, I'm out.
Okay, thanks, Adam. Can you hear me okay?
Yes.
Okay, I'll share my screen. If I can find the right one. Okay, can you see my screen okay now?
I can see, yes, I can see your web browser. Yeah, perfect.
Okay, brilliant, thank you. Okay, thanks, Adam. So my name's Sarah Donaldson. So I'm part of the ensemble HAVANA group. So I work with Toby, Jane and Adam. So today I'm going to go through some like live demos using both the two browsers that Adam mentioned, so the Ensemble Genome browser and the UCSC browser. But just before I do that, I just want to show you this page. So this is the page that Jane mentioned, and this is the Ensemble training page, and this is the page for this this meeting, and this is where we will store the presentations and also slides, sort of screenshots of the demos as well. So because it may be, it's obviously difficult to maybe follow along with the demo at the same time, so you can do this in your own time later. So again, if you have any questions, please put them either in the Slack channel or in the chat, and one of us will answer them. So the first first browser we're going to use is Ensemble. So this is the Ensemble homepage. You can see that there's various different species here, human, mouse and zebrafish, but there's lots of different genomes that you can look at if you want down here. We also, if you're not familiar with using the Ensemble browser, there is some great help and documentation page here. So if you click on this, there's tutorials and there's information on how to add your own tracks and data, and lots more information as well. So this first region I'm going to look at is a predetermined region that we've chosen in human. So I'm going to just click on human here, takes the human homepage, and then I'm going to just paste in some coordinates into this search box here, and this should bring up this page here. So the top box that you can see along here is like an overview of chromosome 16. So this is the P arm and the Q arm, and this red box here shows us this region that we're looking at in more detail below. There's some what are known as assembly exceptions here, and these will be important in one of the other examples that I'm going to show you in a minute. So this box here shows you the genomic sequence, and then it shows you the genes that are on this chromosome, and the direction that the strand of the gene is on is shown by an arrow here. So we can see some of the different biotypes that we spoke about. So there's these gold genes here. And there's these red genes, and these are all protein coding genes. The reason that they're different colours is because the gold ones are where there's an agreement between the annotation from the automatic annotation from Ensembl, automatic pipeline, and the annotation from the manual annotation from the HAVANA group, and the red ones are annotation from one or the other, but mostly from HAVANA. There's also other gene types here. So in this kind of purpley grey colour, these are RNA genes. So this one, for example, is a long non-coding RNA, link RNA here. And this one over here is a small microRNA gene. In the panel below, we can see more detail about each of these genes down to the transcript level. So for example, if we focus in on this CENPN gene here, we can see that there's 7 protein coding transcripts. There's a process transcript which doesn't have a CDS. And there is also a transcript which is subject to nonsense mediated decay, which Adam mentioned, and which I'll talk about a bit more in the next example. One thing to note in Ensembl is that this blue bar here across the middle is the gene. The genomic sequence, and the genes above are on the forward strand, and the genes below are on the reverse strand. And we can see on the reverse strand here, there's a number of different link RNAs that are visible, as well as these protein coding genes. There are actually pseudogenes visible too, but there aren't any in this particular region that we're looking at. So the other thing we can do is we can look at this same region in the UCSC browser. So this is the page for human for UCSC, and we can paste in these same coordinates. Hopefully this will load okay. And now one of the differences between UCSC and Ensembl browser is that all the genes are shown together. They're not shown like above and below the genomic sequence. So the way you can tell which strand they're on is the way the arrows point. So if the arrows point from left to right, it's on the forward strand. If they point from right to left, then it's on the reverse strand. Now the default Gencode annotation that you see in UCSC is Gencode version 32 here, but we can actually see the newer versions of Gencode. If you go down here to this genes and gene predictions section, click on show, and then you can we can look at this in a bit more detail here. So this is the latest version. It's in Ensemble version 34. If we click on this, I want to make sure that we can see both the basic and the comprehensive transcripts. We can put these to set these to different levels. So here I'm just going to set these to pack and then click submit. So here's this same gene here that we could see before, this CEPN gene. The colouring of the transcripts in UCSC is different from in Ensemble. So red transcripts here, for example, these are retained intron transcripts. And if you just click on them, it brings up this information, these information boxes. The green transcripts are non-coding, so these are the process transcripts, etc. And the blue ones are both the coding transcripts and also NMD transcripts, which is slightly different from, again, slightly different from Ensemble. And if we zoom into this CEPN gene, we can see the this is the NMD transcript here for this gene that we could see in the Ensemble browser. So I'm going to move on to a second example now. So this is looking at NMD in a bit more detail. So I'm going to have a look at a gene called SNRPB. So I'm just going to paste this name into here and click go. And then click, hopefully, on the top hit here. And this takes us to this gene. So we can see this gene is on the reverse strand because it's below this bar, and so it runs from right to left. So this is 5 prime and this is 3 prime. We can see that this gene has 3 transcripts which have been annotated. These 2 are coding, and this third one here is nonsense mediated decay. And we can see a bit more information about this at the top here. We can also see the location of the gene. It tells you how many splice variants, orthologs, etc. So there's quite a lot of information that you can get about this gene from here. Now, the reason that this gene, or sorry, that this transcript here is subject to nonsense mediated decay is because it contains this premature stop codon in this exon here. So the filled in boxes in here are where the CDS is. So this represents the truncated CDS in this transcript. And any stop codon which is more than 50 base pairs from a downstream spliced donor site will be subject to nonsense mediated decay and degraded by the cell. So the reason that this NMD pathway exists is to prevent truncated proteins from potentially damaging the cell. So this is a type of translation fidelity mechanism. So the reason that this is, has NMD and these 2 are not is because it contains this extra exon here. This is called a cassette exon. And we can actually have a look at this exon in a bit more detail by zooming into this region. So I'm going to go to click on this region in detail link here, which takes us back to this window that we were looking at before. And we just need to wait for this bit to load. There we go. And if we, whoops, sorry, just have to wait for it to load again. Okay, so if we zoom in here, click jump to region. Let's switch that off for a second. We can see this exon a bit more detail. So if we zoom in a bit more, and hopefully we can get to the base level. And we can see at this point here, so TAG here is the code for a stop codon. We can also have a look at the translation of this sequence. So if you click on this little cog icon here, it brings up lots and lots of different tracks that you could switch on to have a look at in different categories: sequence and assembly, genes and transcripts, messenger RNAs, proteins, etc. So there's lots of information here that you might want to have a play around with and have a look at. But the one we're going to look at today is this translated sequence here. So if we click on that, it fills the box. Click the tick in the top right hand corner, and hopefully this should load up the translated sequence. And here we can see this red box with the star is the stop codon for this particular transcript and this is in the second frame. So you can see all 6 frames of the translation. So these 3 on the forward strand, these 3 on the reverse strand, and this is the one that we're interested in here. Another thing that I want to point out here, if we just zoom out a little bit. Maybe a bit more than that. So there's this track here called this 111 way GERT elements, and if you click on it, it tells you that this is their conservation score. And this is based on the alignment of 111 Eutherian mammals. So not surprisingly, where we see exons such as here and here, we see a score for this. It shows you the score and the p-value if you click on it. But maybe, slightly surprisingly, we also get a hit over this cassette exon. So this suggests that this exon, well, it shows that this exon is conserved, and the fact that it's conserved suggests to us that this has a function. So even though we can see this from here that this is conserved, we might want to have a look at this in the other species that we look at in Gencode, which is mouse. We can do this if we look in the UCSC browser at the same gene. So hopefully it's still there. And click on the top hit here. And we can see this gene here. And this is the exon with the premature stop codon. One thing that you can do in UCSC, which can be quite useful, is as this gene is on the reverse strand, we can actually reverse complement the sequence by clicking reverse here, and then it will take it onto the so that we've got it sort of as if it's on the positive strand, so running from left to right. And sometimes it can be simpler to look at genes in this way. So to view this, so we're currently in human, but to view this in mouse, we can go down here and click on one of these comparative genomics tracks. And the one we're going to look at is this placental chain net track. So if we click on that, it tells you that this is a non-primate placental mammal genomes alignment. So if we switch this on, click refresh, and we can see that this track is here. We click on this, it brings up another window, and we can click on this link here, which is to open the mouse browser at the corresponding position, which I've done here. And we can see, so this is the latest mouse Gencode information here, and we can see indeed that this exon does exist. And if we zoom in on it, we should be able to see down to the sequence level And if you actually look, so I've had a look at this, if you actually look at the exact sequence, there's only 2 amino acid differences between the human and the mouse sequences. So it's very clear from looking at this that the gene organization of these 2 genes in human and mouse is very similar, and specifically that this NMD cassette exon is present in both species. And there is actually substantial evidence now to show that some NMD transcripts may have a regulatory function, and they do this by modulating the amount of protein that a gene produces. So if you find that splicing favours the inclusion of this exon, this means that more transcripts are then sensitive to NMD and are broken down by the cell, and the result of this is less protein is produced. So this is the way that the gene can be down-regulated post-transcriptionally. And it's actually interesting to note that the mutations near this conserved exon are actually associated with a human disease called cerebrocostomandibular syndrome. So the last example that I want to look at is talking about these polymorphic pseudogenes that Adam mentioned. So these are genes where they are a pseudogene in the reference sequence due to either a SNP or an indel, but in other individuals, or say if you're talking about mice in other strains, this gene is actually fully functional and is translated. So the gene we want to look at as an example here is SERPINA2. And so I'm just going to paste that into this same search box that we use for the other examples, and click on the top link here. And we can see here that this gene has appears to have just 2 exons and is labelled as a polymorphic pseudogene. Up here, under this location, there's a link which says view alleles of this gene on alternative sequences, and this is one way of linking through to what I'm going to show you. But I just wanted to have a look at this region in detail again. And we can see that this region that we're looking at comes under one of these assembly exceptions that you can see above and below, that, you know, across the chromosome here, and also by the colouring excuse me, of this sequence here. So this assembly exception is also available or also visible as a track down here, and if you click on the red bar and then click compare with reference, this hopefully will bring up another window, there we go, where we can see these 2 assemblies side by side. So the top one is the alternative assembly and the bottom one is the reference assembly, and our gene of interest, this SERPINA2, is down in on the right hand side here. So clicking on in the alternative assembly here and clicking on the gene name will take us is another way of getting to this page here. And so we can now see that this is actually a 5 exon gene, and that this has this functional CDS. So obviously, if you were studying this gene, this is, you know, it would be really helpful to have the coding gene sequence available in the browser like this. And we can do the exact same thing in the UCSC browser. There we go, and this top one here. And again, this appears to be this 2 exon gene. So to view the alternative assemblies here, this comes under this mapping and sequencing and alternative haplotypes. So if we set this to full and click refresh, then at the bottom here we can see the alternative haplotype, and you simply click on this and then select view corresponding position range on the alternative assembly. And here again, there we can see the full gene. What's also useful in this browser is that we can see that this is the deletion in the reference sequence. So this is the reference sequence now shown at the bottom, and these two lines here represent the deletion. So obviously these are the two exons that we can see in the reference assembly, and this is where these are the exons which are deleted. Okay, so that's the end of the first demo. So I'll stop sharing my screen and I'll hand back over to Jane.
Right, I'm just going to see if I can share my screen properly. Just find my talk in the right place. Okay, here we go. So I'm going to share this screen and hopefully this will work. And I will. So can you see the correct version of the slides, please, Sarah, rather than the
Yes, I can.
weird version of the slides, if you know what I mean?
Yeah.
Sorry,
yeah, not the presenter tools.
Thank you. I have two screens, and even if I don't want presenter tools, it puts it on the other screen, so apologies. Anyway, thanks very much, Sarah. That was great. So I'm now going to talk to you about some of the latest projects and the latest data that we've been using in Gencode gene annotation. I'm Jane Loveland, and I'm also a member, obviously, of the Ensemble HAVANA team. So Anne talked a lot about exactly what we do in Gencode and how we create our transcripts. But what is interesting for us is actually trying to find new transcripts. So if you remember, Adam was talking about how actually the numbers of protein coding genes is slightly fluctuating. And this is because we are actually looking at things which are quite tricky now and things which don't necessarily obey the existing rules of what we think should happen in the genome. So we're trying to use different approaches to try and solve these issues. And we're doing this by looking at functional potential, on the significance of the gene, and then a combined approach with everything. So something for which we're targeting functional features. This is an example of the TJP2 gene, which is involved in eye disease. And this little box that we have over here, we found a couple of extra exons, and these were actually found by looking at phylo-CSF data. So it was revealed to us in this data that there are two extra exons up here, which had previously not been annotated. And also very interestingly, when we looked at the CAGE data, so this is looking at the data at the 5' end, we found that this is tissue specific to retina and also fetal eye as well, which is incredibly interesting when this is a gene involved in eye disease. And also we found that there was a SNP, which could be incredibly important in the diseases associated with this gene. So this shows that by actually adding these new data types in, we can reveal quite a lot of extra information about the annotation that we present in Gencode. So this has led to several clinical projects. This is an example of a publication where we've been looking at some genes of clinical importance which evolved in infant epilepsy. And we have in fact updated 191 genes. And when we started doing this annotation, we had just under 2,000 transcripts, but afterwards we'd more than doubled the number of transcripts to almost 5,500. And what we found, in fact, that we were adding many alternatively spliced transcripts to these genes, and we changed the mean number of transcripts per gene, alternatively spliced transcripts per gene, from 9 to 28, which is a huge amount. And it means that we have a massive depth of annotation now in Gencode, which is incredibly useful on a clinical basis. So this is one of the ways that we can add to the gene set. And in fact, what we found that by adding this extra information, we can also identify putative causal variants, which is very important in disease. So an example here of this SCM1A transcript, we found that by looking at these alternative transcripts, and in fact, we found a potential causal variant, which has been found in patient data. So this is very exciting that we can actually use our annotation, and this is involved in Dravet syndrome, which is a rare type of very severe epilepsy. So this is incredibly useful in diagnosing disease. Another project that we've been working on recently, due to the current reason why we're all not in lovely Barcelona and we're having this remotely, is we've been looking at some COVID-19 associated human genes. So these are genes which are believed to be associated potentially with this virus, and these are associated in various different aspects. We found these from lots of different sources. You can have a look at our Gencode genes page, which gives you a bit of information, and we also have a blog about this to explain what we've been doing. To date, we've looked at 280 genes, and we've modified a huge percentage of these, and we've added several thousand transcripts. And in fact, one of our genes, we've added 114 extra transcripts. So it's quite a challenge for us to keep our annotation up to date, and this is something that I'll talk about a little further. So this is an example of where you can see the annotation. We have an update cycle, which is every 3 months through Ensembl, but we also have track hubs. So if you look for this track hub, or you just search the track hub registry for COVID-19, you will see our latest annotation, and you will see all these different transcripts that we added just in this subset of genes. When we do a full update and a full annotation update, this will actually get incorporated into the Ensembl properly and into the full Gencode gene set, so you'll be able to see all of them then as well. And some of the things that we've actually found as well are, this is an interesting feature. So this is the ACE2 gene, which is the entry receptor for the SARS-CoV-2 virus, and it's a very important gene. And we've actually found, if we look at this top box here, that we found some extra transcription start sites, in particular in lung tissue, which we hadn't found before. So we found some extra exons. If you see here, we had some unchanged models, we've modified some, and a lot of new models as well. So we hope this can be really valuable to have this incredibly deep type of annotation to help, particularly in clinical research. So we know that we have a lot of data, and long reads are really changing the way that we annotate. So by long read, I mean PacBio, Oxford Nanopore, or SLR-seq. And we've seen an explosion in the data types. This slide here just shows you an example of the MAPK10 gene. We looked at some new PacBio data, and these are all non-redundant, and we found 192 alternatively spliced transcripts. This is just a screenshot of what it looks like in the UCSC genome browser. So if we look for more and we add more data, we're always going to find more. So we're a manual annotation team. How do we deal with this? How do we enable us to capture this complexity and depth of sequence in an efficient way? So one of the things that we've been looking at, apart from doing particularly sets of genes for deep manual annotation, like we did with the epilepsy genes, we actually have a pipeline called TAGENE, and this is a semi-automated pipeline which we're using to support the manual annotation. And what this does is this takes long read data such as PacBio, Oxford Nanopore, SLR-seq, and it filters and merges and clusters them into loci. And we then have been manually reviewing as we go through to check whether or not we think that these are sensible. We've then been using short read RNA-seq as well to confirm the intra-exon boundaries, which we found very valuable and incredibly useful. And then adding this to our annotation set, but only when we are completely happy that we think this is absolutely spot on and incredibly accurate. So we've manually checked over 2,000 of these genes already, and we've been using these for long non-coding RNAs, and we have a lot of new loci available now that you can see. What we want to do now is actually expand this. So Adam was talking about the Gencode basic and Gencode comprehensive gene sets. In the comprehensive gene set, we have quite a lot of partial transcripts, and then we're hoping by using all the new data sets, we'll be able to properly expand and complete these partial transcripts by using this TAGENE method. So this is work in progress for now, and you'll see more of these transcripts in the coming months as we use this method in different ways. Another aspect of long read data is actually opening this up to the community to see how computational methods for transcript identification and quantification can work. So we have this project called LRGASP, which is Long Read RNA-seq Genome Annotation Assessment Project. You can see the link here. If you go to the Gencode Genes website, you will find there's this link to LRGASP. And this is actually a project which we invite people to take part. using long-read sequence technology, PacBio and Oxydanaport in human, mouse, and the manatee genome. There's a bit of an outlier in there to try and characterize the strengths and weaknesses of this challenge of using long-read data and actually evaluating different methods for the accuracy of transcript detection and quantification. So if you're interested in that, please have a look on our website, or obviously you can ask us in questions later on. So, as well as adding to the data set and increasing the depth of annotation that we have, another thing that we're very keen on doing is harmonizing our annotation. And I'm going to talk about two different aspects of that, looking at some collaborations with the NCBI, which is the RefSeq team, and also the UniProt database. So to start, we'll talk about refining the human gene set. So the human gene set has supposedly been completed for quite some time, but as we know, the numbers aren't stable, and we're very keen to see if we can find a way of stabilizing them. We've had an existing collaboration with RefSeq since 2005, where we've been working on the CCDS project, which is a consensus coding sequence project, which just looks at the CDS of human and mouse protein coding genes. And we have agreement for many of these, and many of these genes actually have more than one agreement. But what we found that actually, because there's two different sets of human annotation, people either use one or another. So you tend to have people that always use RefSeq or people that always use Gencode. And because our annotation sets are different, because we use different algorithms and we use slightly different ways and different rules in the way that we do our annotation, it makes it hard for researchers to exchange data or translate coordinates, etc., if you're not using the same data set. And people often say, why are the things that you do different? It's good to have differences, but it's also good to try and find agreement on this. So what we're also looking at as well is that lots of different websites such as Nomad, LRG, Decipher, etc., all have their own supposed canonical sequence. So this is their sort of best choice. So what we want to do, and what we've been working on for the last couple of years, is identifying a representative transcript that captures the most information about each protein coding gene in human, not just the longest or the first one. When people are faced with 200 alternatively spliced transcripts in a gene, they're either delighted because there's so many, or they go, which one should I be looking at? So the way that we're working in this project is that we actually want to revise our RefSeq and Gencode annotation sets to match exactly in overall splicing, CDS, and precise 5' and 3' boundaries. So we can create a common gene set which can be used for lots of different applications. So this project is the main project, which is matched annotation from NCBI and EMBL-EBI. And this set has the following attributes. It matches DRC H38, and we have one main select transcript per locus. And these are 100% identical between RefSeq and the corresponding ensemble. And they will have the same 5' UTR, CDS, and 3' UTR. And there is no new identifier. This will use RefSeq NMs and ONS T transcript IDs, which are versioned and matched. And these transcripts should be well supported and conserved and representative biology at this locus. Obviously, we know that we are not saying that you should only ever look at one transcript, and all of our transcripts should be considered, but it's a really good starting point if you want to have a look at a locus and you're not quite sure exactly what's going on. And really importantly, is extremely useful for clinical work. So I'm just going to talk you through some of the stages that we go through to make these decisions. So we start off by comparing transcripts which have been annotated independently by RefSeq and Ensemble. And we have a different pipeline at each site. So RefSeq have their own pipeline, Ensemble have our pipeline. We look at things like conservation, expression, etc., etc. And it runs through, and in a best case scenario, it will pick out an identical CDS and splicing, but you'll have different UTRs. So a lot of people say, well, that's no big deal having different UTRs. But it is actually, if we want to actually match, we need to make sure that the entire transcript matches. And this isn't actually very easy to do. We had to come to agreements on which data we're going to use. We're using actually cage data, the 5' end, poly-A-seq for 3' end, and agree on guidelines of how we interpret this data, because we've been annotating for a long time, both different groups, and we have our own ways of doing things. But for this project, we had to try and create some agreements. So what we've done is for the 5', I'll just show you how we do this at the 5' end and the 3' end of genes. This is the KNG1 gene. This is just a snapshot from the Ensemble browser. You can see that we have here a Ensemble transcript, which is ending here. And we have a RefSeq transcript, which is ending here. We're looking at the RNA-seq, and we can see that we have this peak going up along here. And we also have CAGE counts. And CAGE is really useful to get the full 5' end of messenger mRNA. And we're actually looking at these individual counts, which give us a quantification of the RNA abundance. And you can see these peaks. And when we look at these peaks, we can see the strongest peak, and we can see the longest peak. And actually, we've agreed with thresholding that we're actually going to go for what we call the longest strong. So this is an agreed position that we're going to clip the RefSeq transcript and the Gencode transcript to, so that we have agreement. We have a similar way of looking at things in the 3' end. This is a snapshot of NCBI's genome data viewer. We have a RefSeq transcript ending here in the green bar, and an ensemble transcript ending here in the blue one. We have some INSDC data, which is cDNAs and ESTs, so this is the end of the peak of that data. And we have RNA-seq, and also with that we have poly-A counts. So this is data from the 3' end, from the polyadenylated region of the mRNA. It defines the end of the transcript. And where it's red, it means that we have our highest threshold and our what we think is the highest and best cluster. So again, we have the longest, and again, we have this concept of the longest strong. So it means that we will both clip back our data sets to actually match this position. So this means the coordinates of our matched transcripts are identical. So how are we doing on this? So we started nearly a couple of years ago now. Initially, we got around 50%, which was great, looking at all the genes, and we also have a separate set of clinical because we're interested to see how we can use this for clinical research. And we found actually that we've crept up, but fairly slowly. So our current version, which sadly isn't, I haven't shown it on the graph, is actually version 0.91, where we have 80% of all human protein coding genes have a main select, but importantly 96% of clinical genes, and we are nearly up to 99% now of clinical genes. And the reason it's important for clinical is that we want to use the main select for a clinical use case to help standardize clinical reporting. So we've generated a list of genes from different sources, things like the Transforming Genomic Medicine Initiative, different disease panels, Panel App, ClinGen, etc., etc. And we've been working to actually get these transcripts adopted by key resources. So you will see these in the variant of predictor in Ensemble, and you'll see these actually in Nomad and also ClinVar as well. You'll see that you have a main select transcript, and you will have the Ensemble transcript ID and the NM versioned IDs, which are now available. So this is great. We're progressing really well on this. But what do we do with the remainder of these genes, and why are these ones so difficult? So the ones which are proving quite hard are the ones which have different coding sequences. So these are really complicated genes that have many multiple transcripts. We already know when we've been looking at new sets of data that we can have many hundreds of transcripts per gene, and we'll find that they often have a really similar score using our pipeline. So the only way that we can actually do this properly is by using manual curation, and this takes time. We're also reviewing our pipeline components and seeing if we can actually add different sets of data, and if there's a way that we can improve the pipelines in any way to help us make these decisions in an easier way. We also have differences in annotation. So annotation is a snapshot in time, and that's why annotation can be different from one source to the next. We may have annotated something more recently than somebody did, say, at RefSeq. So in some cases, the RefSeq pipeline may have picked a transcript that's not in Ensemble, and also vice versa, where we have picked a transcript which we think is a bet that's not in RefSeq. So we're actually running our pipeline now on each other's data, and we're having lots of different conference calls to try and actually find convergence on these. So hopefully in the next few months, we will have much higher percentages of coverage. So that's what we've been doing with external collaborations, but we've also been looking at things more closer to home for us, which is UniProt, which is also part of the EBI. So this is the protein database, and we have this website called GIFTS, and GIFTS is a mapping between all of the human and mouse proteins in UniProt to all of the protein coding genes in Ensemble. And you can have a look at this website here. It's just the EBI website /GIFTS. It's just in beta at the moment because it's fairly new, and we need to update our versions, which we're working on now. And we've had a bit of a pilot project where we've looked at around 1,000 protein coding genes in human and mouse. And we've looked at where we can update these, where we've updated them ourselves in Gencode. We've had conversations with Uniprot where we've altered sequence, and some of them we've actually removed where we don't think they're protein coding. And it's important to remember that protein space has been around a lot longer than genomic space. So a lot of these proteins won't necessarily match the reference genome that we're looking at, which is why we do find that we have some differences. And also we have differences in our definitions of what we think in some cases are protein coding genes. But this all adds to our functional annotation as you see things in the website. And then just finally, I'm going to talk to you about some very new work where we've actually been looking at alternative open reading frames. This is a POLG gene, which is very important in mitochondrial disease. This is the canonical transcript, which has 1,240 amino acids, and this is its structure. It's recently been found that by looking at the 5' end of this gene, we have a completely alternative frame of translation. Ordinarily, we would look at this and think, well, this is only 261 amino acids long, it can't actually do much. We would go for the longer one, but what we've actually found that there are signals which have been conserved in phylo-CSF in the different coding frame to actually say that there is something happening here, and this is actually something really important. And also, really importantly, we found that there's a ClinVar variance in there, which could potentially be related to disease susceptibility. And there is a recent paper that you can see here if you're interested in more of this. So this is the kind of depth that we're going into to look at individual genes to try and add yet more interest and more functional annotation to our gene set. So I'm going to stop here and I'm going to hand over now to Toby, and he's going to demo some of the later bits and pieces that we have on the website associated with our new work. Are there any questions that I should potentially answer now, anybody, or shall we continue this in the Google Doc?
I think continue it in the Google Doc.
Lovely, because I couldn't see the questions as we came up. So thanks very much, everybody. We'll move on to Toby now, who's going to do another live demo.
Yeah, hello. So my name's Toby. Obviously work with the three people that just presented before me. I am going to demonstrate some of the features that Jane's just gone over, and to do that I'm going to attempt to share my screen like that, and then I'm going to move that over there. Try to hide. Oh God. Right. Anyway, there you go. Right. Go on. It's there. So the first thing I want to do is demonstrate one of the update track hubs that Jane mentioned. So the ensemble release cycle works on every 3 months, which means that a new release only ever appears every 3 months, quarterly. And so that means that a lot of our most recent annotation doesn't become visible and available to everyone for quite a considerable period of time. So to sort of attempt to partially get around that problem, we have a track hub available where you can see the most recently updated annotation. So this is released every night, UK time, and so any fresh annotation that's been made is visible. And then to demonstrate that, I'm just going to look at a gene that I know has recently been annotated, and that's the TLK2 gene. So I'm just going to search for it in this box here. I'm going to select human because it's human I'm interested in. Click OK. Go to the TLK2 gene page there, and then I'm going to look at the region in detail as Sarah demonstrated earlier, like this. And then I can configure this page on this little sort of cog button here, which allows us to upload and configure a vast number of resources that Ensemble has available. And actually, one of which is the custom tracks, because at the moment the update track isn't available through Ensemble's default list of available columns and tracks. So I can do this. I can go to this track hub registry search here. I can search in Gencode update, and you can see that this loads up three separate hubs, one of which is the COVID-19 gene annotation that Jane mentioned earlier. And the one that I'm going to be using right now is the Gencode annotation updates hub. And so to just attach this, I can click on this button here. And after a little while about thinking about things, it's loaded up successfully, and I can click on the check button there. And after a little while, about thinking about things, finally, up it pops. So this is a view that Sarah was demonstrating a little earlier, where you can see the current comprehensive annotation here, where we have maybe about 15, 16 or so annotated transcripts here. So the red and the gold are the protein coding ones. The blue transcripts are the processed transcripts and the nonsense mediated decay. You can see that this new track that has popped up here has sort of almost doubled the number of available transcripts for this particular gene. And if we were to look at this in more detail and we wanted to see what the ensemble gene ensemble transcript IDs were, we could go to this cog button here and click on the structures with labels. And then after a little bit more time loading up, you should be able to see that we've added a lot more alternative splicing structures throughout this gene in a range of across all our different types of biotypes. And hopefully in a second you'll be able to see the ONS TIDs sort of listed along here. There we go, just like that, thankfully. So you can see now these are completely displayed with their full labels, with the gene symbol here and then the ONS gene ID across here. We can do a similar thing in the UCSC browser. This time I'll use a different gene that I know has been recently updated, this TUB GCP2 gene. Click on it like this. We can see we've got the default Gencode v30 version 32 displayed here. We've got the NCBI RefSeq genes identified here. You can see they've currently got 3 different protein coding transcripts available for this gene and 1 non-coding transcript. And in the current Gencode version 34 comprehensive, we've got maybe 5 different protein coding transcripts, a couple of retained introns, and some non-coding transcripts here. So to look at the full detail of what's recently been annotated, we can add that track hub to this browser by going to click on my data menu option here, track hubs. I want to stay there. And this is the list of publicly available track hubs that anyone can connect to. This particular instance is you need to add in the URL that leads you to the correct web file. So I can just copy and paste this update track hub address into this bar here. This address is available in the resources that you'll have been given access to. So now I need to write it down, and I can click on add hub. And in a second, I should be redirected back to the, should have been redirected back to the gene, but I can just search for it again, like this. And finally, up pops here in these red transcripts of the most recently updated Gencode annotation. So we can do like a quick sort of compare and contrast against what we currently have in the most recently released version of Gencode here, what RefSeq NCBI have here, and then you can see by using the PacBio and SLR RNA-seq long read data, we've been able to add a significant number of new alternatively spliced transcripts to our annotation, which will be released in the next release of Gencode, significantly more than doubling easily what we currently have available. The next thing I want to move on to is an example of new attributes and biotypes that we've recently added to our gene set. To do that, I want to look at the MPZ gene. So I'll just type that into this search bar at the top here, click return. And what we've known for quite a while, but what we haven't had until quite recently is the knowledge that there's a subset of protein coding genes that display what's called stop codon read-through. This is where the translation machinery comes to the canonical or the known apparent stop codon, but in fact doesn't stop there and actually scans through it and carries on translating through that that codon to a further downstream stop codon. And we've known about this for quite a while, but we haven't been able to display it and show it in our annotation in Ensembl. And we've recently sort of corrected that issue. And we can see that in this MPZ gene, well, hopefully we'll be able to see that we can see the main select transcript here, which is 206. We can see that displayed here. We can see that the filled in gold boxes are where the CDS start here and the end here. And we can see there's this alternative transcript here where the filled in box representing the CDS actually extends beyond what's seen in the select transcript. And this is because we've been able to annotate this stop codon read-through feature, which I can demonstrate by looking at this transcript in more detail by clicking on this link here, where we can see that we have this annotation attribute here, highlighted as stop codon read-through. And when we look at the sequence information for this particular transcript, which we can do via these links up here, when we look at the protein sequence, we can see a protein sequence here. I've got the start, and so the alternative colors are the exons. So this is the first exon, the second exon, third exon, etc., etc. In the last exon, which is indicated by this stretch of blue here. Oh, what have I done there? Didn't want to do that. This stretch of blue here, we can see this X here, which would be the stop codon that's used in the main select transcript. But we know from experimental evidence that this locus, this gene, has this read-through, this stop codon read-through, and we've been able to annotate it like this, and therefore extend the CDS to the next downstream stop codon. Another thing that Jay mentioned earlier in her talk was the TAGENE pipeline. This is where we take our recently sequenced and available long read data, and the pipeline, this is fed into the pipeline, and the pipeline produces a number of the annotated models for us, which we can then manually check before they go into our pipeline. So obviously, one thing we wanted to highlight with this pipeline is to flag up which transcripts are a product of it and which ones aren't. And so to do that, I'm just going to demonstrate that using this this link RNA, which is as yet presently unnamed. So it just takes the gene symbol and the gene name is just based on the contig that it's based in. There's a couple of things I want to point out about this link RNA. Firstly, as we look at it here, we can see that it overlaps in close proximity to this protein coding gene, this POU3F3 gene, and we can see that it overlaps, and we flag up that fact with another annotation attribute here where it says overlapping locus. We can see that there's a link to the definitions by which how we describe that and what we mean by that. So that's here. Essentially, it just means that the two loci, as the name suggests, are overlapping. And if we were to look at any of these transcripts, so I'll just take the top one as an example, we can see that the annotation method used to produce it is flagged up as the TAGENE pipeline. So this is defined as a manually supervised computational pipeline that we've used. It takes the long read data, and there's a sort of a description. If you click on the link, you'll be able to find out more about it. So this is just to allow our users to know the methodology and the means by which we've produced these, and these are all flagged up. And at the moment, we've released a very large number of non-coding genes using the TAGENE pipeline, and we're currently working on using the TAGENE pipeline to add and integrate new models into our protein coding genes. And I will again highlight this is all with manual oversight and supervision, and anything that we're not 100% certain of will be manually checked and verified before it makes it into our annotation data set. The last example I just want to go over briefly is the main select transcripts that Jane also mentioned. And to do that, I want to look at yet another gene, this time the FOXP2 locus. And by clicking on that, so we can see here at the gene page, we've got quite a large number of transcripts annotated here. The vast majority of which seem to be protein coding. We've also got 5 NMDs and 4 transcripts. And as Jane said earlier, we often get users sort of wanting to know, yeah, but you know, come on, which is the real transcript, which is the one I should concentrate my time on? And you know, that's sort of given rise to this project we've been doing in collaboration with the NCBI, whereby the 2 annotation groups select the main transcript as the one that they deem the most sort of biologically relevant and functional. And this is highlighted in these flags up here, and you can see that there's this main select button or flag, which when you hover over it gives you a brief description of what we mean by that. And we can see that it's represented by the FOXP2200, and we can see that that is can't see it. Which one is it? I can't even. Ah, it's this one here. There we go. So this transcript here is the one that's been selected based on, you know, a number of features: conservation, strongest cage, cluster, where the poly-A-seq is available at the far end. So this is the one that's been deemed to be selected as the main select, and that's the one that you'll be able to look at through the main annotation. And with that, that's me. Oh, the other thing I wanted to there's also a track hub where you
Toby, you're muted.
Am I muted now?
I'm muted now.
Good. So I don't know how much you missed, but it probably wasn't very exciting. I'm just going to demonstrate the main track hub that's available in UCSC, so you can see the main sequences in UCSC, main transcript, sorry. So I've gone to the same gene, the FOXP2 gene, and I can go to the track hubs section again, and there's another URL for the main track hub. It's this again. This is available in the documentation for this session. I can add that hub in exactly the same way that I did for the Gencode updates. Annoyingly, take me back there again. But by going back to that FOXP2 gene, we should be able to see. Here we go. So we've got the Gencode version 34 annotation down here. We have the NCBI annotation here, and we've got the track hubs that I've just loaded up. The main. So we've got a representation of the refseq NM sequence here. These are exactly the identical sequences, but just from the 2 different sources. This is the NCBI. This is the ensemble one with the ensemble ID here, and it represents the transcript down here. And so this is the one that both groups have agreed upon as to be the main select transcript for this particular gene. And with that, that is the end of my demonstration. And so I'll hand back to Jane or Adam.
Thanks, Toby. Excellent. Right, so please go ahead and add more questions to the spreadsheet. I'm just going to quickly share my screen. Hopefully I've done the right one. So just before we draw this to a close, just a couple of bits of acknowledgement. Obviously, there is a large team of people working on Ensemble projects, so we need to reference them. They're the people that put all of our data on the website and maintain it for us and do our releases. And these are all our different funders. And then these are the specific Gencode people as well that we have. So this is our team. These are the TGMI people who are working with us on the main project, other particularly key people in Ensemble, and the rest of the consortium as well. And at the bottom, again, you can see that we have a couple of links. We have the link to if you just go to training.ensemble.org, you'll see all the different events there, and you'll see all of our information. And then also the Q&A document is down here, which is also posted into the Gencode Slack channel.